Hello dear students, welcome to the class of Eukaryotic transcription. So what is transcription? It is the process in which a new mRNA called as messenger RNA is being transcribed or synthesized complementary to a DNA strand or a gene. So that process is called transcription, and the technique of transcription or the steps of transcription in prokaryotes has already been discussed earlier. So we will just have a comparison, have a comparative look at what all differences are there between eukaryotic transcription. We will see all of them. So we go to eukaryotic transcription. Though the steps, the basic steps are the same in eukaryotes and prokaryotes, the eukaryotic transcription is considered to be more complex, and we will go to the two basic differences between eukaryotic and prokaryotic transcription processes. First thing is that in prokaryotes, we saw that there is an enzyme called as RNA polymerase. It catalyzes the formation of messenger RNA from complementary DNA. So there is only one kind of RNA polymerase in prokaryotic cells, whereas in eukaryotic cells there are three distinct RNA polymerases that transcribe different classes of genes. So that is the first difference. When prokaryotes have only one type of RNA polymerase, the eukaryotic RNA polymerases are of three types. Second is, in prokaryotic transcription, we saw that the RNA polymerase directly binds to the promoter sequence of the gene to be transcribed. That is, there is a hollow enzyme in which sigma factor is also a part, and we have discussed the subunits of the RNA polymerase enzyme in prokaryotes with two alpha, a beta dash and an omega subunit with the sigma factor will directly bind to the promoter sequence of the gene which is to be transcribed in the case of prokaryotes. Whereas in eukaryotes, the eukaryotic RNA polymerase will not directly go and bind to the promoter sequence, but before that it will interact with a variety of additional proteins to initiate transcription. So interaction with other proteins is required for eukaryotic RNA polymerase to start the process known as transcription. Now, so it was already mentioned that there are three types of RNA polymerases in eukaryotes. They are type 1, 2 and 3. So these are the three types of RNA polymerases. First one, we go to RNA polymerase 1. The RNA polymerase 1 in eukaryotes is located in the nucleolus of the cell, and it is assigned to transcribe the 5.8S rRNA, the 18S rRNA and the 28S rRNA. Now we go to RNA polymerase 2. It is RNA polymerase 2 that will transcribe the protein coding genes which will give rise to mRNA. So it is RNA polymerase 2 that transcribes the protein coding genes and forms mRNA. It has other functions also, that is the formation of small nuclear RNAs and some of the small cytoplasmic RNAs. Then comes RNA polymerase 3. RNA polymerase 3 transcribes the tRNA genes. So we can say RNA polymerase, it is with the help of RNA polymerase 1 that these rRNAs are produced. RNA polymerase 2 produces mRNA, and RNA polymerase 3 produces tRNA as well as 5S rRNA. So our focus is RNA polymerase 2, which will transcribe the protein coding genes, thereby giving rise to the mRNA or the messenger RNA. Coming to RNA polymerase 2. So as earlier mentioned, for RNA polymerase 2 to initiate the transcription process, it has to bind certain proteins which are called as transcription factors. It cannot alone initiate transcription. Now coming to those proteins which are called as transcription factors. There are two types of transcription factors. One, the general transcription factors or GTFs, and the other additional transcription factors. So from the name itself, general transcription factors can be called as global. It is there for all the genes, all the eukaryotic genes. So it is part of the basic transcription machinery, and it is involved in the transcription from all polymerase II promoters. So for all genes this is required, that is the general transcription factors. Whereas the additional transcription factors, you can say it is customized, it is different for different genes. So they bind to DNA sequences that control the expression of individual genes and are thus responsible for regulating gene expression. So they are different for different genes. You can say they are customized, whereas general transcription factors are global. Now we will go to examples of general transcription factors. The examples are TF2A, TF2B, TF2D, TF2E, TF2F and TF2H. What do you mean by TF2? TF2 can be expanded as the transcription factor for RNA polymerase II, because it is RNA polymerase II that is assigned for the production of mRNA in the cell. So the transcription factors which assist it can be abbreviated as TF2, that is transcription factor for RNA polymerase II, and these are the examples TF2A, B, D, E, F and H. Now what do? They will bind with RNA polymerase II at the promoter to form a complex which is called as the pre-initiation complex. So for initiation to commence, for initiation of transcription, the RNA polymerase II has to necessarily bind to these transcription factors. It is applicable to all genes, that is why they are called as general transcription factors. Now before moving on to the process of transcription in eukaryotes, we will just have a retrospection of what are the eukaryotic genes. So we know that there will be a promoter, there will be an operator gene or a regulator gene, then the structural gene, and there will be the end. So as we already know, it is monocistronic, there will only be a single gene. A single gene will have a unique promoter for it, and the genes are, they contain both coding regions called as exons and non-coding regions called as introns. And once from these genes the mRNA is produced, the introns are to be removed. So here you can see all these colored bars can be considered as exons, and this interspaced regions, these intervening regions can be considered as introns. So once the mRNA is formed, it will be having all those regions, but it will be later processed to form an mRNA which contains only the exons. So this is the entire process of eukaryotic transcription. Now we start with the first step, that is promoters. That is the first region of any gene which is to be transcribed. Compared to a prokaryotic promoter, the eukaryotic promoters are larger and they are more complex. Many eukaryotic promoters contain a particular sequence. So it is characteristic of many eukaryotic promoters, a characteristic sequence which is called Tata box. What does it mean? Just look at this G. This is the promoter. This is the place where transcription has to begin. So this is the transcription start site and this is the promoter. In most eukaryotic genes, it has been found that there is a sequence which reads as TATAAA, which is around 20 to 30 base pairs upstream from the start site of transcription. So we usually use the terms upstream and downstream. What is upstream? Before the gene in mention. What is downstream? After the gene in mention. So this is lying upstream of the transcription start site. This is the place where the actual transcription has to begin, and this Tata box will be present 25 to 30 base pairs before this start site. So the sequence is called as Tata box, and it is read in the 5-3- direction of which strand? The non-template strand, because this is the non-template strand. This is 5- and here will be 3-. The other is the template strand, where this will be 3' and here will be 5' and the mRNA is made complementary to this template strand, but this is the non-template strand. So, the sequence TATAAA is read in the 5' to 3' direction on which strand? The non-template strand. Okay. So, this can be considered as a consensus sequence which is there in many eukaryotic genes, and this is the most extensively studied promoter. Actually, about 10 to 15%, only 10 to 15% of mammalian genes contain Tata boxes, while the other genes contain other core promoter elements, but it is Tata box that has been well studied and characterized. That is why we are focusing on it. So, this is regarding the eukaryotic promoter. So, the eukaryotic promoters are characterized by the presence of a Tata box which is 25 to 30 base pairs upstream from the start site of transcription. Now, so here again you can see the promoter with the Tata box. This is the transcription start site. Now, the first event is, so it was already mentioned that for initiation the RNA polymerase II, it cannot itself initiate transcription, but it has to bind to transcription factors. So, and it forms an assembly which is called as the pre-initiation complex or PIC. So, we will see how it is formed. So, we already mentioned the names of the transcription factors TF2, A, D, E, H, F, B, etc. not B, etc. So, who is the first to come at that site? It is TF2D, TF2D, and there is a subunit of TF2D which is called as TBP. It is called Tata binding protein. So, the first transcription factor to come and bind here is TF2D, whose subunit is called as TBP or Tata binding protein. So, it is Tata binding protein which will recognize the Tata box of the promoter. So, as a step towards transcription, this TBP, which is a part of TF2D, will come and recognize Tata box. So, that is the first step. Now the other transcription factors also come and bind. 5 more transcription factors, which are they? TF2A, B, then E, F and H, and then the RNA polymerase II also joins. So, 5 more transcription factors and RNA polymerase will combine around the Tata box in a series of stages and thus forms the pre-initiation complex. So, this is a stepwise process we can say, where TBP is the first one to recognize Tata and bind to it. Thereafter, the other factors will come into play and the RNA polymerase will also join them. Now, here you can see one factor which is called TF2H here. This is the TF2H. It has got enzymatic activities also. It has got a protein kinase activity by which it can phosphorylate RNA polymerase II. A particular region of RNA polymerase II can be phosphorylated by the action of TF2H, which is having protein kinase activity. And it has also got helicase activity. It has also got helicase activity. What is helicase? The ability to unwind DNA. Because this DNA has to be unwound, then only transcription can begin. So, TF2H has got helicase activity which will help in separating the DNA strands of the promoter. Then only the polymerase will be able to start transcription, then only the polymerase will be getting access to the template strand. So, TF2H is peculiar in that way. That means it is having a helicase activity by which it is unwinding the template and the non-template strands and thereby helping this RNA polymerase II to initiate transcription. Now, so transcription has begun. This is the DNA, this is the pre-initiation complex. Okay, transcription has begun. Once it begins, all of them move forward except TF2D. You can see TF2D is remaining there itself. Others are moving forward. Others are released from the complex. Now, as long as TF2D remains bound to the promoter, Additional RNA polymerase molecules will be able to attach to this promoter site and they can initiate more rounds of transcription without any delay. So, that is why the TF2D is staying behind, because as long as it is there, other RNA polymerases can also join and they can also initiate the transcription process, so that many rounds of transcription can be done without delay. So, this is all about initiation. Now, we have to go to elongation, but before that we will just mention mRNA processing. So, suppose this process is over and you are getting an mRNA. This is the mRNA, the mature mRNA, immature mRNA. This has to undergo processing before it goes for translation. There are 3 steps. One is a cap is added at the 5' end. Second is a tail is added at the 3' end. So, 5' capping and 3' tailing. A poly A tail will be added and the introns will be cut off, and finally you are getting the spliced mRNA which will be sent to the cytoplasm for translation. So, once the mRNA is formed from transcription, it has to be processed in 3 ways to become the final transcript. Then only translation can begin. Then only it will be exported to the cytoplasm. So, the 3 steps are a 5' capping, a 3' tailing, which is done by polyadenylation, that means A will be added, and removal of the introns and the exons will be joined. So, this process is called as splicing. Now, why it is mentioned here? The transcription process has to complete, no? But we have to understand one thing, that once initiation is over, elongation will begin. Along with elongation, the capping process will also begin. The splicing process will also begin. So, for that I will take you to the next slide. So, I told you there is an overlap in the proteins involved in elongation and those required for RNA processing. That means this mRNA is processed by certain proteins which also take part in elongation. So, you can say they are taking place hand in hand. So, do not think that only after the entire transcription process is complete, then only the splicing will begin. It is not like that. There is overlap in these processes. There is an overlap in the proteins involved in elongation and those required for RNA processing. So, we can say elongation, termination and RNA processing are interconnected and they are properly coordinated. So, I will give you some examples. There is an elongation factor called as SPT5. It will also help in 5' capping. Then, there is another elongation factor called TAT-SF1. It will also help in splicing. So, that is how we can say the elongation, termination and RNA processing steps are somewhat interconnected. They are properly coordinated. Then, now we go to the elongation process. So, once initiation is done, the next is elongation. That means the RNA molecule is slowly, progressively produced by adding of ribonucleotide phosphates. So, thereby the chain is growing. It is almost the same as that in prokaryotes, but a little complex. Why? Because you know that in eukaryotic cells, when they are not dividing, the genes exist as a mass which is called as chromatin. So, it is looking like, so it is in the form of a thread which is called as chromatin. And how this chromatin is made? It is made with the help of such structures which are called nucleosomes. So, these form the units of a chromatin. What is a nucleosome? It is actually a structure formed by 8 proteins which are called histones, and the DNA is coiled around this histone octamer. This model is called as a nucleosome. So, DNA is such tightly packed. The DNA is tightly packed around charged histone proteins at repeated intervals. That is how it looks like in eukaryotes. These DNA histone complexes are called nucleosomes. They are regularly spaced, as you can see, and each octamer, that means 8 histones together, is called as an octamer. Each octamer is bound by DNA that is around 146 nucleotides long. So, this one octamer is surrounded by a DNA which is almost of length 146 nucleotides or base pairs. So, this is how eukaryotic DNA is arranged. So for transcription, there is one more hurdle in the case of eukaryotes. There is one difficulty, that is the DNA is now associated with the histones. This association is to be broken, at least temporarily broken, for transcription to be continued. So for that, there is a protein complex which is called FACT. It is a facilitator of chromatin transcription. What does it do? It is a part of transcription machinery. It will move histones away from the DNA template every time it comes across a nucleosome. So when it moves along the gene, when it is seeing a nucleosome, it will remove the histones. And once mRNA is synthesized, the FACT will replace the histones to recreate the nucleosomes. So that is also important. So this step is not required in prokaryotes because there is no such arrangement of DNA complexed with histones, but it is there in eukaryotes. So that is one difficulty that this RNA polymerase or the enzyme faces while transcribing eukaryotic genes. That is, the DNA is associated with histones to form nucleosomes. So for removal, there is a protein complex called as FACT. It will remove the histones, assist in elongation. Thereafter, it will replace the histones. So we go to a slide where you can see. This is RNA polymerase. This is the gene, the DNA which is getting transcribed, and you can see the histones. These colored blocks can be considered as histones. Histones are H2A, H2B, H3, H4, etc. each in 2 copies. That is how an ectomer is formed. 2 H2A will be there, 2 H2B, 2 H3 and 2 H4 will be there. So altogether 8 proteins will be there. And you can see here, this is the FACT. FACT is actually a dimer which is formed from 2 proteins. One is called spt16, the other is called ssrp1. So this is the FACT. So what is FACT doing here? FACT is removing the histones. See, FACT is removing histones. Okay. So it is dismantling or it is disorganizing the nucleosomes ahead of transcribing RNA polymerase, because this RNA polymerase is coming. So before it comes to this region, these histones are to be removed. So that is being removed by this FACT. So FACT always acts ahead of this RNA polymerase. RNA polymerase is slowly coming. This is the main enzyme which is transcribing. But before it comes, the ground is to be made ready, and that is being done by FACT, which is actually a dimer made up of ssrp1 and spt16. You can see here. So it is, what is it doing? It is removing the histones. Okay. Afterwards, when this RNA polymerase moves, suppose the transcription process is over and it moves beyond this region, what will these proteins do? They will again help in reassembling the histones. Look, they are reassembling the histones. It's like a cycle. It is dismantling them and reassembling them. So that is being done by spt16. So that is elongation. And the other things, the other aspects of elongation are the same as that in prokaryotes. So pre-initiation complex is formed, the polymerase is released from other transcription factors, and elongation is allowed to proceed as in eukaryotes. So elongation is taking place, the mRNA is formed here, this is the DNA, the transcription bubble, all you can see. And just as in prokaryotes, the RNA polymerase is synthesizing pre-mRNA in the 5'3' direction. This is the mRNA formed, and this is the template in the 3'5' direction. This is the template. Complementary to this is mRNA produced. We know that just as in prokaryotes, the template is that in 3'5' direction. So that is all about elongation. Now we go to termination. This is also a bit complex compared to prokaryotes. The first thing is that already there are 3 types of RNA polymerases in eukaryotes. They are 1, 2 and 3. But here we focus only on RNA polymerase 2. The thing is that for all the 3, the termination of transcription is different. So here we will only focus on RNA polymerase 2. The problem is that the genes which are transcribed by RNA polymerase 2, that is the protein coding genes from which mRNA is formed. So the mRNA forming RNA polymerase, the genes which form mRNA, which will transcribe into mRNA, they lack any specific signal or sequences to terminate. It's not like prokaryote. In prokaryote, there is a special region called as terminator, but here there is no such sequence. There is no specific sequence which will mark termination. Now what? How will the process end? Just see. This is DNA. So this is only a schema, this is only a representation. Suppose this is the DNA. This is the mRNA newly produced. This is RNA polymerase II. Now, suppose it is this region. This is the end site. So this is the actual end site. Suppose this is the actual end site. In prokaryotes, transcription process will end here itself. Whereas in eukaryotes, where the transcription is mediated by RNA polymerase II, what happens is the RNA polymerase II will continue transcription around 1000 to 2000 nucleotides beyond the actual end of the gene. So this is the actual gene end. But mRNA is produced beyond that, around 1000 to 2000 nucleotides, and that is done by RNA polymerase II. After that, what happens? After that, the transcript will be cleaved. Here, there will be cleaving at an internal site before this RNA polymerase II finishes transcribing. So, as the process is ongoing, there will be a cleaving occurring at the actual end. So, this portion is released. The upstream portion of the transcript is released, and it is this which will further undergo processing and form the final mRNA transcript. So, this is the actual mRNA, but the process here is continuing. So, what happens? There is now the remainder of the transcript which is still attached to the RNA polymerase, which is continuing its action. At that time, an enzyme will come. It is having 5' exonuclease activity. For example, in humans, it is called XNR2. There are different types of 5' exonucleases. Such an exonuclease will come, and what will it do? It will digest the remaining transcript. It will slowly digest the remaining transcript. That is, it will chase. It will go after this RNA polymerase II. And throughout its way, it is removing these nucleotides one by one. It is actually digesting the rest of the transcript, and once it reaches RNA polymerase II, it will digest the entire transcript here, the unwanted transcript, and it will also help in dislodging, or it will also help in pushing away the RNA polymerase from the DNA. Thus, transcription comes to an end. So, once again, there is no, in this gene which is coding for this mRNA, there is no such region called as termination sequence. So, what RNA polymerase II do is, even after the actual end site, it will continue transcribing around 1000 to 2000 nucleotides upstream. As this process continues, there will be a cutting taking place, and who will cut it, that we will see in the next slide. Here there will be a cutting, and this will now go for processing. What happens to the rest? It will be cut off by 5' exonucleases, which will digest this unwanted part of the transcript and release RNA polymerase II from the DNA template. Now, we have one more answer to get, that is, who will cut this region? How is this cutting taking place? So, we go to discuss that, we go to the next slide. This is the mRNA. This is the mRNA transcript. You can see already the cap is formed there. That is why I told you the processing and capping is almost taking place simultaneously. You just look at this RNA. This is the transcript. Imagine this region. This particular region is the actual end site. So, how can it be recognized? The end site of the mRNA will be having two sequences, one upstream, that is before it, and one downstream. So, what are these sequences? So, imagine this is the actual end site. There will be a sequence, a specific sequence before it, which is AAU AAA sequence. So, there will be an upstream AAU AAA sequence and a downstream GU rich sequence. In between lies the actual end site. So you can consider them like flags. AAUAA and GU rich regions on either side of or flanking the actual end site. So they just look like flags. In between is the actual end site. So who will recognize it? There are two proteins. One is called CPSF. The other is called CSTF. CPSF will come and bind to AAUAAA sequence. CSTF will come and bind to GU rich sequence. In between is the actual site. Now, what will the CPSF do? It will cleave or it will cut the pre-mRNA at a site which is 10 to 30 nucleotides downstream from this site, from the AAUAAA site. So, this CPSF will cut this mRNA 10 to 30 nucleotides after the AAUAAA site. So, this is how the cleaving takes place. And this is also, they are also part of the machinery which is involved in adding tail. We already discussed the mRNA formed will have a cap at the 5' end and a tail, a poly A tail at the 3' end. And the enzymes which take part in tailing, they are also or the CSTF and CPSF are also part of the complex which will help in this 3' tailing. So, that is why I told you mRNA modification of mRNA is taking place hand in hand with elongation and termination processes. So, we will just go back to the previous slide. That is, I told you this is the actual end site where cutting occurs. So, here there will be the AAUAAA sequence and here will be the GC rich region. So, CPSF binds here and the CSTF binds here and here will be, it will be cut, and they are involved in the tailing process also, and by that time the 5' exonuclease will cut off all the remaining ribonucleotides off from this unwanted part of the transcript. RNA polymerase II is now free. So, this finishes the steps of transcription in eukaryotic cells.